So the PPTs are downloaded, right?
Yes, sir.
Okay, so good evening all. So we are back to the second week of our clinical research series. Last week we have discussed about how we can actually, design the observational studies and how we can find the gaps in herbal nutraceutical clinical evidences. This week, Dr. Sriharan is going to talk about two of the major things, that is the basic of clinical trial protocol, and the second session tomorrow we are going to utilize AI for protocol drafting. Majorly the scientific prompting is the one which we are focusing on. So based on that, we will be designing tomorrow's session. So over to you, Dr. Sriharan.
Thank you, sir. Hi all, good evening. Today we are going to see about basic of clinical trial protocol. What are the basic and essential things we want to follow while we writing or while we doing a clinical research? How much important the protocol is very important. We want to going to see. For first of all, under the foundation of clinical research, if you doing any research question, doing any researches or doing any study means the protocol is main and essential thing because it is the backbone of the study. Why means we must always follow the protocol. Why in our day-to-day life also. Today our protocol is what? My protocol is teaching to you, taking seminar on it. Your protocol listening to this seminar slide. So wise we should follow the protocol to achieve something in our life. So in clinical research also same things. The achievable protocol which lead to the good research and good research result takes place. So we want to follow the protocol in effective manner. We will see plenty of topic on protocol today. We go to explore a lot of things about by one slide. Why this is very important means our protocol will say that our consistency of our study, how our methodology, our study design and objectives consistently follow throughout the study. So our protocol will reflect whatever studies takes place. So the scientific consistency is very important, is otherwise known as a scientific rigor and regulatory compliance. So our protocol, if you submit anything to regulatory authority body, example, if you're submitting a document related to a research for institution review board or or institution ethical committee. Likewise, you submitting any document, the first document is protocol. The protocol say about your study. What is your study? What is going on? What should be explored? The protocol say what is you. Likewise, in regulatory complaints is very important, and also patient safety. While doing research, the main concern thing is patient safety. We want to achieve the patient safety throughout the study. If anything happen in bad manner, means our institution review board notify us to clarify that and why this happened or what are the precaution you take. Likewise, they will force us to give the reply and query about, they give the amendment to us. So the patient safety is very important. Likewise, multi-dimensional. Before I'm saying this slide is reflecting here. Yeah, the protocol have not like a one-dimensional. It show the multi-dimensional of the study. It say about what are the scientific figure take place in study, what are the study design, what are the rationale you should follow, and it also say about what are the statistical plan and what are the things should be do in trial, how to manage the query. So it deals with multiple dimensions. So it show the it play in delivering the multiple role in single protocol. Here we are going to see about what are the architecture of the protocol. So when we framing protocol, we want to take the mind these 5 core things in our mind. First thing is study objectives. we will see one by one about very briefly. First thing is study objective. Second thing is inclusion and exclusion criteria. Third thing is methodology, and fourth thing is case report form, and final thing is patient reported outcome. This is the core element while we doing frameworking any research about we writing the protocol means you want to consider these 5 things first major. Then alone you will complete the complete protocol. So we go with one by one. The first is study objectives. The study objective is nothing but it reflect your research question. What your research question? So the study objective is divided into 2 types. One is primary objective and second one is secondary objectives. So primary objective is nothing but the primary objective reflect what the exact your research question. For example, if you're doing any prevalence, for example, I'm saying prevalence of anxiety in general anxiety disorder patient and their quality of life. This is your research question means. Your primary research question is a otherwise called as a primary objective is a prevalence. You concentrate about your title reflecting what is your title reflecting. So you concentrating in what main research question on prevalence. And secondary report question is the additional or subtype to the research question. This deals with it will support the study. It will give the validating study. For example, if you're doing any study means accessing any severity, accessing any pain, or accessing any predisposing factor, measuring something you're doing means it comes under the secondary objectives. So primary objectives in a good research, primary objective should be in a single or otherwise twice. So it is lead to the good research. If we put the primary research in a multiple type of question means it lead to confusion in result. In final, we will lead to confusion. So the primary objective deals with only one question. Secondary follow multiple question. It access the pain, it measure the effectivity of the study, quality of the study, predisposing factors. So secondary objective have multiple role, but so primary objective have only one role that reflect the main research of the subject. So while we doing the study objective, while writing the protocol, we want to follow the SMART criteria. SMART criteria is nothing but specific, SR for specific, measurable, achievable, relevant, time-bound objective. Why this is means your objective should be under in a specific manner and measurable. Your research question should be under in a measurable manner. Unmeasurable question should not be lead to a good result. So your research could be under the measurable manner and achievable manner and relevant to your study. If you're doing prevalence of anxiety means you want to take the relevant to the study. You do not correlation between prevalence of anxiety and explain the quality of life in cardiovascular patients. So it became irrelevant. So your study stand with relevant and it under the time bound objectives. Your time bound objective means if you're doing any observational or SAR study means it under the in a particular specific period. It won't follow rather than period. So it lead to the pressure to the researcher and participant also. So it under the time bound objective. These SMART criteria will give the clarity and feasibility to your research. And statistical power. Statistical power is nothing but a SAP, statistical analyzing plan. While we doing the research, our objectives should align with our sample size. So misalign lead to the miscalculation and mislead the research. So it want to align with our sample size. Second, this is the differences between the primary and secondary objective. Likewise, before I'm saying the purpose is main research question, it should be a supporting research question. Statistical power based upon the descriptive analysis. In exploratory analysis is a hypothesis generation. Hypothesis generator is nothing but giving idea. Your research should give idea for a future research. That is the exploratory analysis. Regulatory impact is in primary objective. There should be get regulatory apport firstly rather than the secondary objective. Numbers. Likewise, before I'm saying the primary objective, any study is one is enough. Typically two is more than 2 or 3 or 4 means it will give burden to the researcher and also research also. So avoid too much of number in primary objective. So secondary objective likewise follow multiple allowed. it can access through 5, 6, likewise anything, multiple allow take place. Inclusion exclusion criteria. The inclusion exclusion criteria is the main thing. Is nothing but inclusion criteria follow right person to the clinical research, clinical trial. While we doing any research, a right participant should be take place in research. Likewise, in other word, we say in inclusion criteria fit for a trial. Though the participants who are fit for the trial, they should be included in inclusion criteria based upon our targeted population. What our sample size based upon our targeted population, the characteristic is nothing but demographic, age, disease stages, previous, what are the previous contained. These are the characteristic. Those who are qualifying from this qualifying these characteristics should fit for the trial and allow in the clinical research trial. Exclusion criteria is nothing but filter of the trial. We filtering the person who are not unable not fit for our clinical trial. This is the exclusion criteria. Likewise, some cognitive impairment patients are not allowed because of their impairment. They are not allowed. Some patients have the drug metabolism problem, which reflect the endpoint. So, you want to identify in these inclusion and exclusion criteria first before while doing a research. So these of inclusion exclusion criteria alone will lead the good research in a good manner. Crafting effective selection criteria. Effective selection criteria is nothing but your selection criteria should be overly strict. Overly strict lead to be limited participant. So we cannot attain the sample size. And another thing, broadly also lead to the variability in the result. So we want to consider these things, take in a corrective action manner while selecting the criteria for inclusion and exclusion. We also consider this factor, disease severity, what are the concomitant medication, comorbidity. You also consider, why means it also reflect in study. We want to take the correct participant to our study. Inclusion criteria examples are demographic consideration. Demographic consideration takes place age, what are the age category, gender specification, through from which geography location. While doing research, this is a demographic characteristic. Medical history, past medical history, present medical history, any history of surgery. Also, you want to consider while including a patient. Clinical parameter, what they are, if they have diabetes means they have a abnormal value in blood pressure. If they have a cardiac related any issue, means they have LLD, cholesterol level have be high. So we want to identify the clinical parameter while before we include in their study. And exclusion criteria. The exclusion criteria is unfit for the trial or filtering of the trial. We want to do correct inclusion criteria because the correct inclusion criteria lead to the good and structured research. Why means if we include the wrong person in the trial means it lead to the wrong calculation, wrong sample size calculation, wrong quality assurance. It in the end of the result it affect our study. So exclusion criteria is main thing. Is divided into two types: safety related and study integrity. Safety related means if you are doing study on any drug means renal impairment patient or any renal failure person should be avoided. Why means our drug cannot tolerate to that person, so we want to avoid that person and pregnancy patient, history of cancer having. So these are the things to be avoided. And study integrity. Study integrity is nothing but who are able to be included in the study. Example, cognitive impaired patients are unable to provide the informed consent form. So that kind of the participants should not allow to do in research. These are the things while we consider study integrity in exclusion criteria. Methodology. The methodology transfer our study objective into the procedure manner. So methodology is the very important. It is also our in our body, how the heart is important. In clinical research, methodology is very much important. Methodology show what study design you are taking, what study design fit to your research. Likewise, your methodology say about what study design is suitable for your research question. Likewise, the methodology is very much important. It should be in a clear manner, and and it should be in a correct study design we want to choose. If you're doing any observational study, means you want to, likewise our before class we say, you want to, any rare outcome means you go with cohort study. Any rare expose means you go with cross section study. Likewise, you want to choose. This is the core things in methodology component. First thing is study population. Our targeted population should be included in a right manner, recruited in a correct manner. If we fail to recover right population, right target population, means it also affect the statistical analysis. It also affect our results. So study population is major thing while doing methodology research. So based upon your study population, your research question, your research design should be speak. Likewise, study design. Study design, your study design have two type in clinical research. One observational study, last class we study. Another one is experimental study. If you go with observational, you're doing any observational, no intervention group in your research question, no intervention group is needed. You go with the observational study. If you want to intervene the patient, likewise treated versus untreated, mean you go with the randomization or blending. Likewise, you take your research design. And schedule of event. Schedule of event is nothing but, while we doing the research, we want to collect the data, we want to follow up the patient, we want to track them. So that is the schedule of event. Schedule of event is also otherwise called as a trial calendar. It's trial people calendar. What the things are happen, what the things are, what to do, what are the things want to do, how to do, when to do. Likewise, the schedule of event takes place. Intervention, while we doing any randomization or RCT, means the intervention groups play major role. So this is the intervention group. Data collection. Data collection should be in a quality assurance manner and under the SAP. SAP is nothing but statistical analysis firm. So the correct and accurate data collection will help you to analyze their statistical plan to our study and give the result in an effective manner. So data collection is very important, because during data collection alone, the mislead data collection takes place. Some data collection should be mislead, some data collection should be misreader in the data collection. So we want to follow the accurate data collection using the trained staff. Study design consideration. Likewise, before I'm saying that study design, first of all, study design study. If you are doing observational study, you are going expose, rare expose, go with the cohort study. Rare outcome, go with cross-sectional study. Likewise, you want to choose based upon your research question and your disease status or disease. What are the disease you are going to take? Likewise, your research question and disease nature, you want to choose the observational study. Hybrid design is nothing but it's the combination of observational and randomization. It also very useful nowadays. The majorly using the hybrid design. Why I'm saying means we intervention group takes place, like RCT follow. We follow treated group, untreated group. Observational study also takes place in second part, like data collection and follow up and observing the patient day-to-day activity. Likewise, the observation also takes place in hybrid design. This is a combination of both observational and intervention study. Intervention study is nothing but you all may know about that. It is a experimental study. Here the random RCT follow, RCT the people selected, one in control group, one in placebo. Likewise, you follow the clinical trial. And another one is randomization and blending strategy. Randomization, there are three type of method follow. Simple randomization is a computer generator randomization. We simply allotted to the people, you take this place, you take this, computer generate, and the peoples are included in study. In simple randomization, the one bias what is means, it give the unbiased equal distribution of the control and placebo group. Why I'm saying means, in simple randomization, it a computer generator. So in control group, they have 10 participants, but in placebo group, they have only 5 or 6. So it affect the result and it also lead to the bias. Block randomization, it avoid the balance bias. Why I'm saying means, we dividing the patient into sub block, block 1, block 2, block 3. Block 1 drug control and placebo, block 2 control and placebo, block 3 block control and placebo. They are equally distributed. That is a block randomization. Stratified randomization also another one of type, but it is different to the block randomization. Stratifying means nothing but we are dividing based upon their characteristic. Here the characteristic what mean age, sex, and gender. Likewise, we are characteristic. While we doing research mean 10 male versus 10 female. Likewise, we are stratifying the gender. Using the stratify randomization, we can achieve. And blending approach. Blending approach is nothing but it had under the 3 type. Single blending is no way. The participant unaware about what they are doing, what the research question, they are unaware about it. Double blinding means nothing but the participant investigator are unaware of it. Only the sponsor or the other investigation can know about that. Triple blinding means the participant, investigator, and data analysis unaware that. Only the sponsor alone they know. They simply follow the trial. They know what trial going on, what the result will come to publish. They don't know. They simply follow the protocol. Likewise, our trial protocol they follow and give the result to the regulatory authority. Schedule of event planning. The schedule of event planning is nothing but before I'm saying is a trial calendar. Yes, all the trial, principal investigator, sponsor, CRO, clinical research coordinator, all they have look eye on this schedule of event planning. OE means if we have any error in the schedule of event planning means it will totally collapse the data collection. So schedule of event planning is main important. It will say what and when the data should be collected and what should be collected, and it also useful for prevent the missing data. While we doing a multiple status study means we have a multiple follow-up screening week 1, week 2, week 3, week 4. Likewise, we want multiple follow-up during that week. We schedule of event plan the week, and it will tell to the participant to come and visit the clinic or trial area, and they will note the result and they enter the result, they transfer to the data management team. Likewise, the schedule of event planning is the main thing that plan the data collection and how the result pass through after we finishing the trial design. The trial design pass to from trial design to data collection, management, analysis, and result. The transformation should be helpful using the schedule of event planning. Case report form. Case report form is important while doing a research. Why means if we collecting anything, for example, in your college, conducting any competition, you have a separate form for separate things. For example, poster presentation group have separate form, and another one is what any abstract presentation groups have separate form. Likewise, in college itself, they following the different form for collection. In clinical research, this is very important. This is the report of our trial, because this report say what is data. This report say what is result. This report also helpful for the future trial. So the case report form is very important. In case report form, it should be under the correct font size and correct manner and correct word count and correct box. While we giving doing in case report form also divided into 2 types. In traditional, in past about before 20 years or before 10 years, they follow the data collection through their manually printed form. They collect the data collection form, they enter the value. Nowadays, the most of the research are going on electronic report form. So it is very easy. We also see very deeply in about upcoming slide. In SNTL CRF component, we want first of all you want to take the mind while your CRF form what means. First of all, say about your title and investigator and date. You want to collect every date, because not every date, your date is main important. Which date you collect, which date the people sample size is in this size, it will useful for the statistical concept. So date, and after you following the administration information and go with the demographic detail. And demographic detail say about age, gender, ethnicity, baseline characteristic, what are the things are take place in demographic characteristic, and medical history. present medical history, previous medical history, comorbid, present medical drug, surgery, likewise. In another one thing I also added in this section, and social economy and how the peoples are occupational, what are their occupation. These are main important because we doing any occupation therapy study means their occupation is main important. Likewise, occupation. These are takes place in first phase and followed by next is laboratory data. While we collecting the data like any CBC count or doing any liver related disease, mean kidney function test, depending upon our research nature, the laboratory data change and adverse event. This is main important. While safety concern takes place in adverse event. If we are giving any drug that give any undesirable effect, mean we want to firstly report to the in a adverse event form. Then alone after reporting, we also want to send to the regulatory authority. We will say detail about upcoming slide. Here the design, principal design. Likewise, I before saying it should be in a clarity and simplicity manner. Why means it should be also seen by a patient. Some patient have also knowledge, but they some patient have no medical knowledge, but they will able to know what they are doing, what the study is going on. That able to know. So it should be in a clarity and simple manner and data validation. So the principal core thing is data validation. So in a traditional thing, we should be enter the data. In electronic ECRF, data should be entered in electronic form, and it also auto check the validation. What are the range? It also auto check manner. So data validation. And another thing is regulatory complaint. OI means you will submit the CRF form to the regulatory complaints. Every CRF form. So it also under the ICH, GCH, ER6 in a concerned manner. E6 in concerned manner. This is the difference between electronic and paper CRF. So in previous 10 years back, they are using the paper CRF. Now they are changed to the, most of them are changed to the electronic CRF. So what are the benefit of these are? While doing data entry in electronic CRF, it should be real-time manner, and it's also in easy. We can easily note down it. And in manual means sometimes we mislead or entering takes place. Sometimes it also gone, or we left from some area. So these are the things takes place in entry. We want to entry in a manual way. Query management. While electronic CRF, it automatically alert these queries are in remaining. These queries are want to resolve. But in manual, we will forget how the query last sitting they have said about one query, but we forgot it. Likewise, the forgotness likewise takes place in manual query generation. Data security, it's secure in our app or it under the audit trail. It should be taken care by our who are taking the paper CRF. That person want to taken care of the CRF, because this is main thing. Because this last means our study design gave mislead and it give confusion in the end of the study. So cost, it like nowadays also electronic CRF in free tools also available. But in we doing any high level study means we want to install some software while doing eCRF. In paper CRF there is no cost. It is under the only paper cost. It also taken by the management. Accessibility, it remotely acts. The physical person always carry the data collection form from the everywhere. Data quality assurance. The data quality assurance, nothing but our collecting data should be, we want to verify the data. We want to verify the data. While we doing ECRF means if your BP is 120 to 80 means your range automatically valid. So in ECRF it's also one of the benefit. Manual, there is no auto range check, likewise there is not take place. So data verification is very much important. You want to verify your CRF form, what are the data collector and entry into the trail registry or clinical data. There should be a team clinical data management. You want to enter into the you are transfer your CRF form to them. They will enter their values. And data monitoring. And also in data quality assurance, data monitoring should be very important. Why means it's under the protocol compliance the data takes place. We'll be doing a multi-central trial study means the data collection from multiple trial region. So that is very difficult to collab the multiple trial data in a single trial. So the data monitoring is very much important. And query resolution. If in data collection, some of the queries also takes place while we doing any research, we query should be came from a regulatory compliance or from investigator or anything. We want to resolve the query immediately from qualified staff, which lead to avoid the discrepancy. Patient report outcome. Patient PRO. PRO is nothing but patient voice, or otherwise called as a patient perspective. In about following the previous slides are say about what is research, what are should be followed, and what the research perspective, what the research answers perspective, what the research result perspective. There is no say about the patient perspective. Here we know about what the patient voice and what the patient perspective in their research. Patient report outcome is otherwise called as a patient voice. While we doing research, using this patient report outcome, we assessment what are the things means their symptoms, their function, the quality of the life. We will assessment through the PRO outcome. Is also assessment used by instrument. That instruments also should be in a valid manner and should be we want to apply to the regulatory complaints. We are using this tool and also approved from the regulatory complaints. We alone we want to use the tools. This is a type of the PRO instrument. The selection of PRO instrument is very much important. Why means we want to select the clinical relevant mission to the patient. While we doing a diabetes study means we want to select the diabetes related questionnaire form DSQMS. If you doing a asthma study means you study the asthma quality of life questionnaire form. So you want to choose the specific study for specific PRO instrument to the people. So clinical relevance is very much important. These are the instrument. These instrument health related quality life. It assess the physical mobility characteristic of the patient, treatment static patient, how the patient treatment status together. They will convenience or in a very poor range or likewise patient stratification takes place, and functional ability also day-to-day life exercise mobility takes place, and symptom specific means how their symptoms, severity of pain, and what are the symptoms they are taking, severity or scale using the symptom specific scale. PRO instrument selection criteria. Before I am saying it under the psychometric property. Why I am saying means it should be in a reliability and validity and responsive to our relevant clinical research. Choosing our PRO instruments in a current clinical relevance manner. So our instrument should be previously have a success rate have high. If you're choosing one instrument means this instrument have more than success rate than failure rate. So on you use the instrument. It also helpful for your study to outcome their result. So choices of the right psychometric property is very important. Another one is clinical relevance. Likewise, before I'm saying means your study objective should be in asthma related means you choose the asthma quality life assurance. Your study is based upon cancer means you choose the FACT-G, functional assessment of cancer treatment generic. You choose that. If and also this is the clinical relevance. And next thing is regulatory acceptance. It also accept and approval by regulatory. While we're doing any tool using in clinical research means we want to approve from the regulatory. It's the first main thing. After alone we use the tool to validating their outcome. Common PRO instrument. Before I'm saying this is SF is a short form 36 used for the likewise before saying the general health quality related life, any physical any mobility status we can use it. EQ5DC is a 5-dimensional matter. It involves the economic evolution, physical evaluation, symptoms. Likewise, 5-dimensionally it will give the outcome for the patient. Fact G is nothing but functional assessment of cancer treatment generic cancer patient in trial phase 1, phase 2, phase 3. The PRO instrument used for the patient outcome. VAS pain scale mainly used for osteoarthritis patient. Why means VAS is a visual analog scale. While doing any pain of severity, majorly they use for orthopedic they use VAS scale, and VPAF is a work productivity scale, work productivity assessment instrument scale. Is also useful for any economic analysis or health economic useful. This tool is very useful. PRO data collection. First of all, while we collecting, we want to do pre-planning is very much important. Anything in research also, if you're doing with pre-planning, pre-studying is very important. While what are the things are taken pre-planning means, we want to educate the trial staff how to use this questionnaire form. What are the questions should be asked to the patient, or what should be, how to be collect the data procedure. Likewise, we want to pre-study planning will take place. And the second thing is participant training, and we want to train the participant according to our PRO instrument. And afterwards, data monitoring. After we collecting the data, we want to monitoring any missing data that takes place, or any related to the peer instrument. Data monitoring also takes place. And after data monitoring, we want to analyze what the data gave, and what are the patient outcome through this instrument. We want to analyze the plan. These analyze plans also used for handling the missing data. Now we see about the protocol example. Before we lengthily see about what is research, what are the core element, how are the research CRF takes place, how PRO instrument use. Likewise, we now see what are the example scenery or example of the protocol writing. Here our protocol writing takes place in a herbal medicine observational study. Our title of the study is Prospective Observational Cohort Study Evaluating the Safety Effectiveness of Standardized Passiflora Extract. The passiflora is nothing but Passiflora indica in adult with mild to moderate anxiety symptoms. So, in study title itself, you want to say the primary objective. Easily we know our primary objective is evaluating the safety and effectiveness. So that is our primary objective. Secondary objective is moderating the severity of anxiety or pain or scaling. Likewise, we can multiply choose the secondary objective. Rationally, likewise, we before study, before class, we seen about rationally very deep about very deep in manner and evidence gap. Likewise, rationally deals with our herbal study takes place in traditional use evidence and preclinical data. These are the rationally study evidence supporting the trial. Rationally also very important, because it will tell what are the past history and past evidence which helpful for our study. Likewise, primary objectives and secondary objective we are dividing. Likewise, I am saying the primary objective is a safety profile. We are safety profiling the use of Passiflora over the 12-week we are using. Our secondary objective is using anxiety scale, and we also evaluating the quality of life improvement and dose response relationship, and also identifying the treatment response to the predictive treatment response to the patient. This is the secondary objectives. Participant of selection criteria. Here itself you will get idea what are the inclusion exclusion criteria will come. Likewise, before you seeing that, you will come to the idea of inclusion criteria. Likewise, our study takes place in adult. So adult age group is 18 to 65 years. And also mild to moderate, we previously said our inclusion criteria, and the person who have ability to understand the informed consent form, and they are using the stable current medication, not using any cancer treatment, HIV patient. Likewise, they are not patient, they are a stable patient using the current medication. Exclusion criteria are, we are choosing only mild to moderate. So we want to exclude the severe anxiety disorder patient or pregnancy patient. We will not include the pregnancy in the study. So we pregnancy patient. And allergy to the study help. Some patients have allergy. So allergy patients should be excluded. And they have a psychiatric medication. Some patient in anxiety patient also take different psychiatric drug. Why means they have anxiety plus bipolar disorder, because they have taken different risperidone, haloperidol, so it can make affect or interact our study. So we want to also consider exclusion in psychiatric medication. Study design methodology. Our study design is observational study, prospective cohort design. It's under the 12-week observational period. You know about cohort study in previous slide itself. So it is a 12-week study, and it is a multiple site recruitment. So in here itself, you see multiple site. Why we using multiple site recruitment mean? Because of to attain the sample size. We are collecting the sample from multiple sites. And another one is natural history comparison. Natural history comparison is nothing but our research studies say our result comparison with what already published in a database. So we want a comparison. If we using the supplement, or using the supplement to this treatment, herbal anxiety disorder, have improvement versus the people not use the supplement. Our outcome is our supplement give the high rate, high outcome in the patient. Likewise, we are comparing the natural history comparison. This is a schedule of event, our trial calendar, how the informed consent form, what to do, when to do, our demographic screening and GAD, general anesthetic scaling, quality of life, safety of environment, herb usage dietary. Screening takes place for baseline. The screening deals with baseline characteristic. First, well, we starting before study, this is screening is the first baseline characters. After week 2, week 6, week 12 and follow all schedule using by schedule of event. Case report form for herbal study. Using case report form, likewise we before see the baseline characteristics are age. Here we using the age as 18 to adult age and gender, education, employment status, anxiety history. Likewise, we baseline characteristics should be take place. And additionally, we want to also herbal product usage. Why means we using the herbal product. So we want to say dose frequency, interval, any discontinue, any modification, any relapse. Likewise, we want to take place in herbal products. Concomitant medication. While the patient have any other concomitant disease rather than pediatric psychiatric medication. So, what are the concomitant medicine therapies are take place included in that. Adverse event monitoring, main important. While means we giving any drug, it cause any adverse event monitoring, we want to immediately report that adverse event and immediately send to the regulatory authority. It categorized by different stage. It will be seen in a safety monitoring surveillance in upcoming slide. PRO instrument in herbal study. So we want to, in PR instrument, you want to take in mind 3 things. What main thing is the instrument should be validate and reliability. Second thing is it should be clinical relevance and patient should the participant should be trained to that instrument. So you want to consider these 3 things and you will use the PRO instrument. Here we using the GRD-7 anxiety scale. It should be a clinical relevance and short form total healthy survey also clinical relevance. We using 2 type of primary PRO measurement. Secondary PRO measurements are quality, sleep quality index. Why means the anxiety person have disturbance in sleep. So we accessing the quality of sleep index. Another one is treatment satisfaction. Whether the treatment is satisfying the patient or unsatisfaction takes place in treatment satisfaction. Data collection procedure. It is a rigorous procedure and important procedure while doing a research. After we doing all those things, PRO instrument, the data collection is main thing. Why means we capture the data through the CRF or anything. We want to follow the standardized SOP. Why means if we do not go with SOP means we make fail or we may mislead to do some other way. So all the researcher should follow the SOP. While doing research, data collection procedure, data collection management, have certain, we have the clinical trial, have clinical research, have clinical protocol. Likewise, data collection team has standard operating procedure. Through that standard operating procedure, we collect the data in a correct manner and we enter the data and verify the data and exclude to the analysis. So data collection procedure is a very important thing while doing the research. We are using the electronic capture system means also it very well useful. Why means it will auto range take place and it also catch the error and it also catch the missed data. So using electronic data capture give more benefit in research. Another one is safety monitoring procedure. Now we want to look onto the safety monitoring. The safety monitoring is type. There is a type of safety safety report takes place. First one is adverse event. Second thing is serious adverse event. Third thing is SUSAR. Serious, suspected, unexpected, serious adverse event. These are the type of report. Well, the adverse event you take place mean it under the adverse event. Serious, it is producing any life threatening to the patient or a long time of hospitalization. takes place, serious adverse, suicide, death, lead to death, or it give major life threat. Likewise, we want to categorize the thing, and this safety monitoring are monitoring by DSME. DSME is a safety monitoring data safety monitoring board. They are monitoring. If we receive any adverse event, means minor, means we will have time 15 days to report, and moderate light before light to above the minor moderate takes place, it takes 7 days or something. Very serious thing, major side effect or major thing that lead to the patient life or death, you want to report the regulatory authority immediately from 24 hours. Very much important. We want to report the regulatory authority immediately in 24 hours itself. These are the things are safety monitoring procedure. We want to continuously monitor the safety surveillance, and any adverse event take, means we want to casualty assessment takes place, how this happened, and we want to under the casualty assessment takes place, and safety review, we want to give the report to the safety review committee. They will review and give the feedback to us, or any changes or anything to be moderate, they will notify to through the board. Statistical analysis plan, SAP, this main important. After data collection, safety consideration, statistical analysis and plan is main important. We divided into primary analysis and secondary analysis. The primary analysis deals with descriptive statistics data, the percentage of the study, graph of the study. Likewise, they deals with the primary analysis. Secondary analysis deals with mixed effect model. Mixed effect is model is nothing but we are doing 12 weeks, so there is be a repeated measure that deals with the mixed effect model. And regression analysis in we are analyzing the response of patient. For example, how the response should be in younger patient, how the response should be in older patient. Likewise, regressing analysis and takes place. This also regression analysis also identify the dose relationship of the patient, dose relationship between the dose and the patient characteristic. Quality assurance measure. First of all, we want to train, site training is very important. We want to train our staff according with GCP, good clinical practice, the data collection standard. We want to train the staff. What are the quality assurance should be in the measure, how to follow the audit trail. When we inspection came, how to we will clarify our audit trail. Likewise, we want to train auditors should be takes place while doing a quality assurance measure. The trained qualified experienced auditor we need for audit trail. And monitoring plan, a regular site monitoring plan. We want to access our protocol from up to down, how our data should be going on. In multi-central trial, we have to have a monitoring plan. So these monitoring plan also interlink with the safety, efficacy, and ensure of the patient safety participant. So monitoring plans also take place. Data management. Data management also done by the clinical data management teams. Through our data should be in under the accuracy, consistency manner, any missing data leading the study mislead or any dropout. Likewise, data management should be measured in quality measure, in quality assurance, QA measure. Now we going to see about how the development process takes place, how many months, how many long it will be take. It means we cannot develop the protocol in a simple day or within a 10 days, within a 15 days, possible. So we look onto its timeline is about month, takes place 1 to 8 months. First of all, we want to in first 1 to 2 months we will concept of development takes place. We reviewing the article from database, hypothesis formulations, and likewise we before before class we know how the database, MPACE, likewise Cochrane. Likewise, we concept of development, whatever research question takes place. Likewise, we do the database stage in concept of development. After database stage, we come to the drafting, protocol drafting. We come to the medical writing, or otherwise known as regulatory writing or protocol writing. We come to that point of view, and after that we go to the review and approval. After our medical writing and protocol writing in a finalized manner, between the collapse of clinician, statistician, biostatistician, and a training auditor, we go to the review and approval stage. It also takes 5 to 6 months. In review and approval, there should be a approve, there should be a minor, there should be a approve, there should be a terminate, or there should be notify for any minor changes, or they notify. So review and approval take place 5 to 6 months. After we approve from the institution review board or ethical committee, site preparation is very important. We want to prepare the site and want to put the contract for CRO, clinical research coordinator. We want to put contract, and we want to train the team before initiating the trial. We want to do the site preparation take place. This is the 7 to 8 months. What are the stakeholder collaboration? While we doing this study, what are the collaboration of the team? Clinical investigator. That should be a physician or a chief doctor. Likewise, the clinical investigator takes place. One biostatistician takes place, used for statistical analysis, and one statistician also takes place. And regulatory affair, because we want to do regulatory writing or approval from the we want to need the regulatory executive to get the approval, or you reply to the approval, reply to the query. Likewise, we need a regulatory affair. Data management. Data management, clinical data management want to do, likewise, or otherwise called as the auditor, trial auditor, we know. And ethic export, because ethic rights is very important. So ethic export also, these are the collapse of the stockholder while writing the clinical trial protocol. Common protocol development. What are the challenges? What are the challenges means the main challenges is time and budget. Our studies should be in take inconsistency or in mislead. The main thing is study and timeline. And then another one is regulatory navigation, because we want to follow different regulatory procedure for different country. So the regulatory navigation also much difficult to follow. Well, submission rejected. Likewise, we get submission, we get rejected by reviewer. Likewise, these go in a different country, follow different rules and regulations. So regulatory navigation take place. Patient burden consideration. While we'll be doing the research in a long manner or in timeline constraint, I mean, patient also became a burden to patient. So these are the things, burden, but patient burden considered also takes place. So these are the things are the main challenges. Protocol amendment management. Protocol amendment management is nothing but you are giving for review. They will give the reply of your review, like in a they will give the reply or reject or they approve. Likewise, takes place in protocol reviewing procedure. Reviewer amendment management is nothing but all ongoing trial. There should be any changes from your side. It should be amendment through the protocol amendment management. Type of protocol amendment means administration amendment. You are changing principal investigator. You are changing contact information. You are changing number, phone number. You are changing site. Likewise, that is under the administration. The approval notification only. There is no lead for any ethical committee, regulatory authority to they look on it. And minor means you are changing CRF. Case report form, any changes takes place. Likewise, you are adjusting the patient follow-up. Likewise, it should be in a minor and is expected in a review manner. Review and they will accept it. Why mean because of this minor. Major is mean you are changing inclusion criteria changes. You are including someone or you are excluding someone or any new safety procedure or safety guidelines you are taking to recover the patient means major. It should be in a full committee review, ethical committee, and also review by the DSMP and also review by the DGCA. Likewise, it takes place. Substantially means you are changing the primary endpoint modification. Your primary endpoint is what? Here your primary endpoint is safety profile. You are changing the safety profile into a different manner. If your primary endpoint in diabetic study means HbA1c. So you are totally changing with changing the endpoint to study changing to cardiovascular rate. Means you are totally changing to lipid profile like cholesterol profile. Likewise, you're changing the primary main point modification. But most of the study, they don't do subsistence yield because it also give drawback to our research. So it approved by the our if your study takes place in FDA means we apply the FDA and review committee, ethical committee, DSMP. Likewise, there is a different law regulatory authority. So you cross all the one will be giving this type of amendment. Best practice in protocol development. These are the protocol writing excellence. We want to write in a clear and precise manner. So your writing should not be in a unambiguous language. It should be in a simple manner. It should be give a clarity to reviewer. Your over parliamentary word or over expressing word mislead the trail and also confuse the reviewer. So clear and precision is main important. And another one is comprehensive coverage. You should want to cover all those things from the study objective to the PRO. If any mischange will lead to difficult to get the approval. Regulatory alignment. And also it's under the ICH GCP E6 alignment. Your regulatory alignment should be in a local regulation. Likewise, it under the guidelines. Feasibility focus. How the available resources, sites, qualified staffs, and population you have. The feasibility focus also main things while writing this excellence, protocol excellence. Technology integration in modern protocol. In here we see in a short manner. In tomorrow class we will say we will detailedly see what they are. In technology integration, nothing but we are integrating using like a digital biomarker, wearable watch app, likewise digital technology to implementing the modern protocol to accessing remoting. Likewise, before we see that decentralized trial. Likewise, we digitally going the digitalized technology modern protocol. The digital health consideration, what are the opportunity means? Well, digital health consideration give the continuous data collection while we in a remote manner. So continuous data collection, reduce the site visit, safety monitor in a real-time data manner, and enhance the patient engagement very well in a patient. I mean, we accessing in remote alone. So challenges means during technology there should be validation problem takes place. Sometime malfunction or repair device it give the misvalidation lead place. And data privacy and security, because of some country follow the different IT rules. So some research they should be do not allow this kind of the tools should be affect the individual privacy. Likewise, data privacy and security takes place. And digital divide consideration means while inaccessible patient, while the older patient they have no accessible. Likewise, they take place. And regularly acceptable patches, because we using these kind of the tool means we want to accept from the regulatory authority. So it also under connect with IT Act, Information Technology Act. So we want to follow these are the things. Global protocol consideration. While we international level global consideration, cultural sensitivity. Why the cultural sensitivity takes place means the different country follow the different cultures and procedures. So the culture sensitivity takes place while doing the international protocol consideration and regulatory harmonization. Different country have different rule. FDA have different rule. EMA have different rule. Our CDSCO have different rule. So regardless takes place. So regulatory harmonization, localized language. If we giving study in any Chinese government, Japan health and welfare means we want to localize to their language. So these are the considerations. In India, there is English alone takes place. In Russia or otherwise Chinese means they will majorly their forms are in their national language. Chinese, like with Chinese, Russian. So language localization is takes place. Risk management and protocol development. While we doing protocol development, there should be any research, there should be a risk should be exposed. We want to manage the risk. That should be in the 5 category. First of all, identifying the risk. After identifying, we validating. After validating, we maintaining the we taking the control measure. After control measure, we again reviewing risk mitigation plan taken place, and lastly we go to the review. These are the risk management plan. After this risk management plan, we will list the critical risk from 1, 2, 3, 4 by following orders. So this also takes place while any deviation or anything in the protocol, the risk should be arise. So this is the risk management category from 1 to 4. Future direction in protocol development. Using the AI, we can develop the protocol development using prompt. Likewise, we see detail in tomorrow class. Real world evidence using the ECRF, electronic case to perform, and it also used for the precision medicinal approach using biomarker, digital biomarker they are using. This is the future direction in protocol development. And this is the quality metric of protocol excellence. Your protocol should be in excellence, mean it under the protocol adherence. Your protocol should be adherence in 95% and should be under the minimum 5% of query rate, not greater than 5%. And your patient retention and dropout control is under the 90%, and now your type of amendment frequency also in lesser than 6%. These value tell the quality of matrix of your protocol. So this is the quality matrix in complaint from the protocol adherence, quality rate, rational rate, and amendment frequency, followed by these 4 following is major important while calculating the quality matrix. Professional development in protocol design. While we want to develop more while we want to much into in regulatory writing or much into medical writing, means we want to develop our skill. We want to go for the medical writing intensive or class. Likewise, we want to go workshop. We want to go and learn about what is scientific writing, communication. And while we regulatory writing means we want to knowledge about ICH guidelines, their quality guidelines, safety guidelines, and efficacy guidelines, and METRA guidelines. Likewise, we want to knowledge about regulatory knowledge. While we going with the statistical analysis, we want to know about statistical methodology, likewise study design, likewise, and project management capability, operation management, how our management takes place, our capability takes place while leading the team. Likewise, operation management, we want to gain skill from operation management, like course, and clinical research ethics. Doing study, know about what are the ethics, what are the GCP guidelines, what are the notification from the ICH. Likewise, we want to make essential knowledge on these, which will helpful for to write the protocol design, or we writing go to job like any medical writing or regulatory writing. Continue education, nothing but going a workshop or doing any intensive with medical writing or regulatory writing, or regularly follow up the regulatory guidance update, and therapeutic area specialization. If you are a clinical pharmacist, you want to take the therapeutic area specialization, because if you are in any nephrology ward, you should be in focus on in kidney research. Likewise, BCP, likewise there are major course takes in therapeutic area specialization, and technologies and advancement training, likewise AI integrating, and international conference participation. Using international conference, we are publishing our abstract article, which will give look on to the coming person to what are our professional development. While we go to any interview for the medical writing intern or regulatory writing, this will give the weightage for our included to job or selection of the job. This will give the weightage to that. Today, these are the key takeaway. You may know about the patient-centric design, what should be a participant-centric design, and you also know what are the collaborative excellence while doing the protocol, and quality by design, how the quality range from while doing the protocol excellence. These are the takeaway from your in today basic clinical trial protocol. Excellence in clinical trial protocol. This is the example for the well-designed protocol which lead to the meaningful clinical research. So first of all, you want to design well-designed, which will lead to the success of the clinical research. And remember one thing, every protocol represents the opportunity to contribute the medical knowledge. So your protocol should be in medical knowledge, and it also improve on the patient outcome. So your approach should be majorly focused on medical knowledge and patient outcome. Thank you for the opportunity.
Thank you, Dr. Sriharan. There are a couple of questions.
Okay,
so
sir.
the first one is, how do protocols differ for various phases of clinical trial?
Yeah, why means the phase one takes place only the predetermined dose of the drugs. So it's follow different protocol. In phase two, it should be in patient, first in patient. In phase one, it deals with only in healthy volunteer. But phase two is different. So we including the patient participant in the trial. So the phase two also different. And phase three is the confirmatory trial. It's tell about the ratio of risk benefit and what are the safety concern takes place. And phase 4 is post-marketing surveillance. So the protocols differ from various phase.
Okay. Next is what statistical method and data management plans are part of the protocol?
Statistical method is nothing but you all may know about the power, statistical power. Using that statistical power, we using SAP. While we doing a statistical method means we want to follow the statistical analysis plan using the biostatistician. They will give the plan. They will give follow the certain standard operating procedure. Likewise, they will do. In data management, there should be a statistician. So, or otherwise also known as clinical data management. It also nowadays B.Pharm, PharmD or graduates also takes place under. Using that, we will look for to how to collect the data included in the protocol. We will see on it.
Okay. And last question is how protocol ensure consistency in multiple clinical trials?
Yeah, this is a nice question. Why means the while we doing the multi-center trial means our protocol should be mainly concerned on data management. Why means we are collecting data from the different trial region.
Different.
So there should be a inconsistency takes place or mislead takes place. So these protocol ensure that consistency from multi-clinical trial. So while we doing a multi-center clinical trial, protocol play major role so ensuring the consistency of data.
Okay. Thank you. Thank you, Dr. Sriharan. It was nice
Thank you, sir.
presentation.
Thank you for the
Tomorrow we will
opportunity.
meet at the same time and we will discuss about using AI for protocol drafting, where we will be using scientific prompting as one of the tools. So thank you all the attendees who were connected. Thank you.
Thank you, sir. Thank you for the opportunity.